Hi everybody, welcome to Forestry Optional and welcome to the lecture 2 of paper 2B wherein we talk about forest ecology. Okay, so in the previous lecture I think we've completed the very basics, biotic, abiotic factors, forest community concepts, right? Succession, climax, productivity, nutrient cycling, etc. Now we will talk about identification of species, dendrology, taxonomic classifications, things related to herbaria and arborata, and then in the third part, okay, we will talk about the role of ethnobotany. In this lecture, we will also complete clonal parks also. Now, this lecture is slightly factual and also technical in nature because I will be talking about the various classifications from a botany perspective, right? So here, and I will also tell you what is the most important out of this, okay? So for example, when I tell you the different classifications, within that, the most important one will be the Bentham and the Hooker classification, okay? So when I'll tell you, so initially if you do not have a base here, you might feel because there is a lot of classification and a lot of things to cram, okay? But again, when you once you revise it once or twice, this will not be that difficult of a chapter. And almost every year you will see questions from the classification or at least herbaria, arborata, etc. I think in 2021 they even asked you holophytes, neophytes, all these different different terms also, okay? So I've also added a lot of previous year questions here also, so you will get to know what are the kind of questions which are asked. So we'll start with dendrology, okay? Again, this is logy, so of course you have to read or study about something. In this, what you are doing or studying, this is the science of studying trees, okay? So dendro means trees, logy means to study. So this is basically the study of trees, dendrology, okay? So again, these dendrologists are just like your detectives who study the trees. They study the characteristics, relationships, and they then try to understand it in a deeper manner. That also helps. Again, for example, when I tell you bamboo or when I say sal, shorea robusta, within that you can also have some other species, etc. So we need to understand dendrology and we need to understand different species for a better understanding, right? So first thing is that why do you need to have dendrology, and then what is the role of dendrology in forestry? Okay, so first of all, the very important thing here is identification of trees. Again, in this lecture itself, when I tell you herbarium, when I tell you clonal parks, arboreta, etc., you'll also understand that how to identify. Okay, these are just like people can also make some journals. Okay, you have reference books. In the similar manner, we can also create recognition based, let's say, journals, etc. So basically dendrology first of all helps you to know what are the trees. So you see the flower, you see the bark, and you see other attributes of it. So number one, that is going to help you, right? Then it is also helping you understand the tree relationship, okay? So how different are the different tree species, how they are interacting, etc. All of this can be known, and I will also give you examples as to how this also is, let's say, understood, right? Then the more you understand about something, then you know what is the soil pH, temperature, the area, okay? So it also helps you in XC2 conservation, okay? So overall conservation can be better done here, okay? For example, when I'm talking of, let's say, red sanders, a lot of illegal trafficking of red sanders, etc. is going on. So if you know about it better, so you can also you can have insights into its growth, distribution and conservation needs, right? So this is Pterocarpus santalinus, okay, native to the parts of southern India, and it is heavily smuggled, okay? So red sanders is one example that you can think about. Then in case of timber production, okay, so here also if you have good knowledge, so again you can also improve your economic yield. You also can understand better what are the seasons, etc., in which something can be carried out, okay? So even that is one thing that is helping you. So role of dendrology in forestry is important. And last is the ecological understanding, right? Again, trees play very specific roles. Again, you can think of how they can help you in soil health. How can they regulate the microclimate? Again, think about 1B and 1A. A lot of these things you can just understand better. So overall, the knowledge of dendrology will help you in many different, let's say, facets. Okay, so that is one thing you can think about. Okay. So here also you can just write a definition additionally. It is the study of principles and procedures for plant identification, as you've seen here, identification, nomenclature and classification. Identification, nomenclature and classification. The principles and procedures involving it is known as dendrology. Okay, classification, identification, nomenclature, etc. of these plants and trees, etc. So that is basically dendrology is a branch of forestry in that sense. Okay, then the case studies I've added here. So one is the case of sal tree. Okay, so sal is very Shorea robusta is important from the cultural as well as economic angle. Okay, however, there are different types of salt tree in the different regions. Okay, so when the dendrologists actually studied it, so they actually, let's say, understood the variations within this one species itself. Okay, so one example can be that the distribution of the single species can have different variations. So dendrology helps to understand that. Then let's say if you want to conserve a species, in this case we've seen the example of red sanders, that is also very much important. And lastly, bamboo. By the way, bamboo I'll do in a lot of detail when I discuss non-timber forest products. But still, you have bamboos, you have Dendrocalamus strictus, etc. different varieties. So then what is the difference between the different bamboo varieties itself? How, let's say, one is to be cultivated, etc. so on and so forth. So dendrology overall helps you in understanding all of this. Fine. So 3 examples we have seen, and just the basics of what is dendrology and what is the role of dendrology in the forestry, right? Then moving one step ahead, because I told you about nomenclature classification. So now we talk about taxonomy. Okay. So this is one thing. So what is this? What is the science of naming, describing and classifying an organism? Now again, if you have read about the plant and the animal kingdom, there are different principles, there are different ways of classification. And through classification, and by the way, a question can also be, why is classification necessary? Okay. So the reason is that, of course, one, you can scientifically segregate a lot of species, helps you in better understanding, better education, better outreach. Okay. So if, and by the way, the whole edifice of science lies on organized classification. Okay. Otherwise, if things are scattered, etc., you cannot properly carry out proper works, right? So that is one thing you can think about in this case, that why is classification or taxonomy necessary? It is because of all these things. This is more specific for dendrology, but overall it is very much important. Okay. So one is what is the definition of it? Okay. Second is what are the types of taxonomy? I've mentioned 3, but there are also other, let's say, specific types. Let's say one is alpha taxonomy, you have beta taxonomy, you have gamma taxonomy. Okay. So alpha will tell you the basic description and the classification. Okay. Beta focuses on the arrangement into the higher taxa like genera, families, and I'll also tell you the whole sequence of this part. Okay. And gamma tells you the biological aspects of these species. Okay. So for again, there is a rank of species. So basically you start with a kingdom. And this will also be when I'll tell you some or show you some diagrams, you'll also get to know. So you have a kingdom, you have phylum, you have a class. Okay. Then you have an order. Okay. And then finally, then you move on to the family, genera, and then species. For this also, you can also create your own mnemonic. By the way, now if you remember, sometimes in mangrove question, they asked you the 6 genera. So genera exactly is here. And sometimes they ask you species. So you start with a kingdom, and then you have a phylum, class, order, family, genera, 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 and species. This is the sequence which follows. In fact, when I tell you that there are this kingdom classification is there, right? Or perhaps when I tell you, let's say kingdom divided into other places. Okay. So we'll also understand this whole part here. So this is how you can just have a basic understanding about what is taxonomy related to. Okay. Alpha, beta, gamma, I think we have understood what is that. Okay. So for example, in alpha, you only talk about morphological characteristics. Okay. Whereas in beta, you go into not just morphological, but also genetic, anatomical, okay, cytological. Cytology basically means the study of cell. So so that is even more. So that is why arrangement of species into higher taxas. Okay. And then you also have taxonomy. Okay. So that is specifically with respect to how the cell, etc. is structured. Okay. There is also numerical taxonomy. Okay. But again, they've not asked you a lot of detail about all these parts. Okay. Numerical is again going into a lot of observable characteristics. Okay. So additionally, two other things I've also told you. One is cyto, one is numerical taxonomy. Okay, so again, many of these types can be known to you. Okay. And I think the sixth one, which is based on the evolutionary, how the evolution has evolved. For that, I will also write the name here. 1 second. Yeah, you can write down omega. Omega taxonomy basically is from an evolutionary perspective. Okay, but how has the evolution been throughout? So taxonomy, what is it? What are the types of it? Roughly should be known to you. Okay, and then this is the part I was telling you, classification. Right, this is basically the whole edifice of this lecture. What is this? It is the method to organize living organisms into, so again you want to group the organisms which have similar traits, okay, and the ones who have differences you just move them out. That is one. Why is this important? Again, understanding relationships, okay, provides a framework for studying biodiversity, okay, more from an educative point of view. So there are many benefits to do that. Okay, now the types are, there is an artificial system, there is a natural system, there is a phylogenetic system. Okay, in all of this, just remember the keywords because this is actually very much detailed. I have also added a mind map. So basically these three are the more main ones: artificial, natural and phylogenetic. And you also have numerical taxonomy and you also have molecular taxonomy also. Okay, now within this there are some famous botanists also who have done their work. Okay, right from, I think I told you, Bentham and Hooker. Okay, there is also the work of Raunkiaer, there is also work of Linnaeus, there is also Eichler. And again, when we read about Bentham Hooker, that also has works of, let's say, Bentham Hooker, Eichler system, Hutchinson system. But out of this, one of the most famous one is Bentham and Hooker. So what you should do, firstly have an idea that we are going to discuss about in classification three basic types: artificial, natural and phylogenetic. Okay, so firstly we'll understand this, and after that we will associate one famous botanist with each of them. That is how you can structure things in your head. Okay, so when I'm talking of artificial, so this is based on the superficial characteristics like a color or a habitat. Okay, and this will also get more easily understood through this mind map also when I explain this. Okay, so just give me a few moments. Then if I talk about natural, it includes internal structures and it includes multiple characteristics. Okay, so that is one. And then you have also a phylogenetic, okay, which is just like omega. So this is based on evolutionary relationships, and after some point of time I will add one name of each botanist, because sometimes they ask you exactly from the name of a botanist that what is this classification related to. Okay, and I think if you were preparing for your environment, at least the plant kingdom or at least this image cryptogram phanerogram is perhaps known to you. Even if it is not, I'll just quickly explain this to you. Okay, so, and in fact that this largely follows the Bentham and Hooker classification, so that should not be a big problem to understand. Okay, so first of all there are three types. Okay, and of course you can also extend it to more types also, but basic three types are artificial, natural and phylogenetic systems. Okay, so first of all I hope this part is clear to you that you have artificial system, you have a natural system and a phylogenetic system. Now previously I gave you this sequence: kingdom, phylum, class, order, let's say family, genera, species. So for example, this particular diagram is very much famous. Okay, so in 1969 you had the 5 kingdom classification: Monera, Protista, plant, fungus and animal, which was then made in 1977. You had bacteria, Archaea, Protista, plant, fungus and animal. Okay, and then you also had some domains. So domains were just like bacteria, Archaea and Eukarya. And then you had kingdoms which had Urobacteria, Archaebacteria, Protista, Plantae. So through the color code you can also understand some basic part as to how the classification was done. Okay, now we will also talk about these parts here. So basically when I'm talking of artificial system of classification, so it is based on one or very few external characteristics. Okay, these are very much superficial and this does not include evolutionary history. Okay, right, so it can be based on a habitat or a stem, etc. When I'm talking of evolutionary history, it is actually the case of phylogenetic system of classification. Okay, second, when I'm talking of natural, okay, so this is based on multiple characteristics. So here one or few, this is multiple. Okay, so again what is the point of difference that you should know? So one or a few, here it is actually multiple. Okay, So this is how, let's say, you can be aware about, let's say, these basic points here. Okay, fine. Then when I'm talking about the other parts, for example, just a second, yeah. So one is, in natural classification, you have multiple characteristics. It is very comprehensive. It considers morphology, anatomy, I told you, all these cytology, etc. All of this is covered in the natural. So here in the natural classification, you have classification of flowering plants based on the flowering type. This is very much important because when I discuss with you Herbarium or Arboretum, sometimes they ask you what is the system of classification which is used. In fact, that is also a previous question. That is why we are discussing all of this. Otherwise, we could have even, let's say, not done this in this much of detail. Okay. So again, I think the first part, or at least this part, should be at least known to you. Okay, that what we are talking about in this case. All right. Okay, just a second. Yeah. So this, please make sure you understand this better. So please put a star mark in the natural system of classification. And I will also write the name of botanist here so that you are able to follow. Okay. Phylogenetic, just remember evolutionary relationships. Okay. So it uses genetics because yes, this is evolutionary. It uses morphology, etc. So this is, first of all, these three terms should be known to you. Okay. Now here I will just write the name of the famous, please pardon me for the spelling if there is something, because I'm writing this very fast. So Linnaeus, we'll also see about Linnaeus. Okay. Linnaeus. So this should be AEUS, Linnaeus system. In natural, I will write here Bentham and Hooker classification, which is the most famous one. And in phylogenetic, we can talk about the this Eichler system. Okay, these are the name of the famous botanist who have classified. Okay, so either they can ask you this name or they can ask you through the name of these botanists. In both these terms, you should be aware what has basically happened. Okay, but out of all of this, the most important will be the natural one through the Bentham and Hooker. And by the way, the Bentham and Hooker model was further even more specialized by Eichler and then later on by Hutchinson. Okay, which I will tell you when I discuss. Yes. So Bentham and Hooker model was there, then modified by Eichler, and then the Hutchinson model, which was fairly recent. And this also, let's say, spoke about monocots and dicots and more specialized. Okay, so this is also one thing you have to know. But again, we will also try to understand what these terms basically mean. Okay, so till here, I hope you're able to follow. Okay. And then there is also numerical taxonomy and molecular taxonomy, but now this part is not that important. Just remember numerical taxonomy. We'll talk about the numerical methods, okay, traits, the variations, and based on that it is going to structure. But again, not that important. Same molecular taxonomy. You think about the molecular data, and based on molecular data, you are able to organize things. Again, this is not that important. Okay, we are focusing on the first three and the names of the botanists who were very prominent. Okay, that is one. Again, talking of classification, you also had Raunkiar's life forms. Okay, please, this should be Raunkier, R-A-U-N-K. Okay, pardon me for the spelling. Who again, the Christian Raunkier was a botanist, and basically he has classified plants based on where they store their buds. Again, a system of classification. Okay, so again, you're understanding as we're discussing dendrology, taxonomy, and classification, that different botanists have used different mechanisms to classify. Okay, so sometimes, and I am telling you that this lecture is slightly factual because these are the kind of questions they are asking you. Okay, so sometimes they may ask you what are the, let's say, Raunkiar's life forms. So basically, the plants are classified on the basis of where they store their buds during unfavorable or wintry conditions. Okay, unfavorable wintry conditions is what you need to know. Phanerotypes. Okay, then you have gametypes, hemicryptophytes. Okay, then you also have this cryptophytes, and then therophytes. You also have few others, but again, these are more prominent ones. Okay, within these life forms. So again, phanerophytes. So tree and shrubs have the buds above the ground. So these are phanerophytes. Okay, again, everything is related to where the birds, etc., are being stored. Fine. Chemiophytes have the birds close to the ground. So let's say this is the ground, the birds are very close to the ground, whereas in phanerophytes, these birds are above the ground. Okay. When hemicryptophytes, the birds are at the ground level. Okay. Crypto, the moment we see the word crypto means hidden. And I will also link this when I tell you cryptogams and phanerogams. Cryptogams means something which is hidden. So of course the birds etc. are underground, so they are not directly visible. That is these are cryptophytes. Okay. Therophytes, again, here the annual plants which are basically surviving as seeds. Okay. Phanerophytes, chemiophytes, hemicryptophytes, cryptophytes, and then the last one basically being therophytes. That is something, just have a look at it. You don't have to exactly cram it, but this can also be asked as Raunkiaer life forms, so please be aware about it. Okay. Okay, before I tell you individually Linnaeus and Bentham and Eichler, I'll just want to tell you this part here. Okay. So for example, when I'm talking of plant kingdom, okay, so plant kingdom can be divided into cryptogams, okay, and they can be divided into phanerogams. Now I'm also explaining this because see, in the current affairs, the India's first cryptogamic garden was in news, which was in let's say Dehradun, Uttarakhand. So again, when I was explaining that in my environment classes, I also explained this whole part, what is cryptogam, phanerogam. So I'll just again, let's say repeat. The name is crypto. Crypto means something is hidden. Okay. So here the reproductive organs are hidden. So of course, what is there in phanerogams? The reproductive organs are visible. That is the first point of difference. The second point of difference will be that in cryptogams, they propagate or reproduce through spores, whereas phanerogams propagate or reproduce through seeds. So first of all, this is the main point of difference between a cryptogam and a phanerogam, that the reproductive organs are hidden in cryptogams. The name crypto tells you. Phanerogams visible. Reproduction is through spores. Here the reproduction is through seeds. One. Second, when I'm talking of cryptogams, here I will talk about three things: thallophytes, bryophytes, and pteridophytes. TBP. In this order: thallophytes, bryophytes, pteridophytes. When I'm talking of thallophytes, okay, so here everything is undifferentiated. And as I move to bryophytes, starting you may see leaves, stems. And when I talk of pteridophytes, it is proper. So the example for pteridophytes is ferns. Okay. So ferns will have proper roots, proper leaves, etc. So the degree of differentiation, undifferentiated to slightly differentiated to more differentiated, thallophyte, bryophyte, pteridophytes, all of these are cryptogams. Similarly in phanerogams, now other botanists have classified it differently. Some of them spoke about, let's say, gymnosperms, angiosperms. Some spoke about monocot, dicot, and then let's say gymnosperms, angiosperms. So that is how this was classified. Okay, so again I'm just repeating this to you so that you understand the kingdom classification. Okay. So now let's say if I talk about here, so cryptogams that does not have seeds, we already know that they reproduce through spores. Okay. In this you have algae, you have mosses, and you have ferns. Algae are thallophytes, mosses and liverworts, bryophytes. Okay. And then let's say, so this has no true root, stems or leaves. Okay. Mosses and liverworts will have some root and leaf like structure. So there is a slightly better differentiation. Okay. Then the third has roots, stems and leaves. Okay. So again, this is how, let's say, this thing is spoken about. Okay. So just a second. Yeah. So that is there. So ferns, horsetails are pteridophytes. These are roots, stems and leaves properly differentiated. This is cryptogam. In phanerogams, if it has no flowers, it will be gymnosperms. It has flowers, it is angiosperms. And this can be divided into needle-like leaves. Think of conifers, think of chir, pine, Pinex roxburghi, etc. Okay, that is conifers. And if it has wide leaves, that is known as, let's say, cycads, ginkgos, etc. So again, if it has seeds, if it does not have flower, it is gymnosperm. If it has flower, it is angiosperm. Okay, and within that, needle-like leaves is conifers. Wide leaves are ginkgos, etc., cycads. Okay, so first of all, please understand this, and through this you will be able to further understand how all this classification was done. Okay. Okay, so that is what we will, let's say, read about. Now let us go back to artificial, natural and phylogenetic. Okay. In artificial, the most famous one is the Linnaeus system, a very famous botanist. Okay, so what did Linnaeus talk about? Okay, a system which classified plants. Again, remember this is an artificial system. An artificial system will have very one or few characteristics. Okay, so here it will talk about plants based on the number arrangement of their reproductive organs, especially stamen and carpels. Okay, again in your basic biology you might have heard about this part here. Okay, so this is what, let's say, they're talking about. Okay, so here it is very simple and easy to use. It is based on the sexual characteristics of the plant. Okay, so Linnaeus Systema Natura classified the plants into, let's say, one stamen or two stamen, because it is classifying them on the basis of their sexual organ. So remember, this is an artificial system of classification. That is one. The natural system of classification, which is more important to us, is the Bentham and Hooker system. I am again and again speaking about it. Classifies plants on broad range of characteristics, and it is not just reproductive. Okay, so contains aspects of plant morphology. It is widely used. Please notice this term, because this is also mentioned in your syllabus, herbaria. It is used in the British herbaria. Okay, and their work is classified into these dicotyledons. So you have polypetali, gamopetali, and monoclinidae. I'll also tell you what exactly this is. This is the Bentham and the Hooker classification. This you should be now able to draw. Okay, I'll just explain this to you also. And then you have the phylogenetic system of classification given by Eichler. Okay, and they have, let's say, this botanist classified this on evolutionary relationship. Okay, talking about non-flowering cryptogams and phanerogams. So this is actually evolutionary. This is Eichler's system. So when you have seen this, think of Eichler. So in your notes also write Eichler here. But again, this is also very much important to understand natural system of classification as well, because this is actually the natural system of classification. Okay, fine. So this has, and by the way, this phylogenetic has recognized 4 divisions like thylophytes, bryophytes, pteridophytes, and spermatophytes. Okay, so first of all, this is what we've understood. Okay, we have seen that there are, again going back to our old mind map, artificial system, natural system, and phylogenetic. Phylogenetic, we will think about Eichler. In Eichler, we'll think about cryptogams and phanerogams. In Bentham and Hooker, we'll think about overall the system, which has, let's say, 3 main groups: polypetali, gamopetali, and monoclinidae. We'll also see what is this. And then the Linnaeus system. So first of all, take some time and absorb what we have spoken about. Linnaeus is the most easy, basically reproductive organs, stamens, etc. So 1 stamen, 2 stamen, how these are getting classified. Okay, fine. Now this is your Bentham and Hooker classification. So plant kingdom, cryptogam, it is lacking seed, and you have phanerogam, etc., which is seed bearing. Now this is divided into monocotyledons and dicotyledons, and in this is the part polypetali, gamopetali, and monoclinidae. Okay, so polypetali is having free petals, gamopetali means used petals, and monoclinidae basically means the flower, etc., which is basically being unisexual. Okay, so again this is one part you can think about. So again in the Bentham and Hooker thing, this is a natural system of classification. Okay, so again that is perhaps one thing you can think about. So again think about monocots, dicots, within dicots 3 different classes. So this is how this entire thing is basically mentioned. Okay, so this is the Bentham and Hooker classification. Okay, and again you also have a particular classification. This also we've understood: cryptogams, thylophytes, bryophytes, pteridophytes, phanograms into angiosperms and gymnosperms, and angiosperms into monocots and dicots. Okay, now this we will also understand through a difference between these 3 systems. So one is the Bentham and Hooker system, second is the Eichler system, and third is the Hutchinson system. Okay, now what has happened here? So basically just remember the main features of all the 3. I know this is slightly more detailed in nature. Usually they ask you this particular system. Okay, but it is also better to know other systems as well. Okay, so Benthamian system, this classified seeds into the spermatophytes. Okay, and the dicots are divided into polypetali, gamopetali, and mono. So remember this poly, gamo, and mono here. This is one of the mone, and monocots are classified separately. Again, see, monocots are separate, dicots are mentioned into 3 different classes. Okay, that is very important. Now, this is widely adopted and used in many herbaria and botanical gardens, especially in the British Empire. Fine. Eichler system is different. Why? Because it is evolutionary based. Okay, it is a phylogenetic system of classification. Now, in this, what has been done? Cryptogams are separated from phanerogams. Number 1. Okay, it is evolutionary from a simpler to more complex system. Cryptogams, thallophytes, bryophytes, pteridophytes, and phanerogams into gymnosperm and angiosperm. And this lays the foundation for the Engler system, which has further refined the plant classification. So again, we are moving into one step ahead. And lastly is the Hutchinson system. Okay, so here also what you have to understand, it is also a phylogenetic. Again, this is evolutionary. Okay, now here angiosperms are divided into dicots and monocots. Okay, angiosperms divided into dicots and monocots. So in this diagram, you should write down, this is basically the Hutchinson system. Okay, I'll also write it here so that people don't get confused. Okay, this diagram can be drawn in Hutchinson system. So there are slight modifications which have happened. Again, the whole idea is to classify it better, to make it so that the people can understand this in a better manner. Okay, so this is how you can understand the different systems also in this. Okay, now we are talking of taxonomy. So now we will talk about what can aid your taxonomy classifications, etc. So in this you have catalogs, manuals. Okay, you have botanical gardens, herbariums, museums, zoological park. So now we want to study about herbariums, clonal parks, and arborata. Okay, so these 3 things are usually asked in your exam, so that we'll see. So firstly, what is a clonal park? Okay, so here let's say imagine a garden in which instead of different types of plants, you have multiple copies of the same tree. The name is clonal park. Okay, so it is basically cloning something. Okay, so here remember this has the same tree. Okay, so here genetically identical tree clones of tree species are grown. This is the whole idea, genetically identical. Okay, and here also if you are taking a single parent tree, so you are choosing a tree which has very good or superior genetic traits and again cloning this out. That is the whole idea. What is the feature of this? Okay, so of course one is uniformity. All the trees are clones, they look and glow similar. Again, aesthetic wise this is better. Okay, and then you can also choose superior varieties. So within trees also, let's say there is one superior variety, you're cloning it, so that can also done. You can also get the best desirable traits. Okay, then clonal propagation allows for producing large number of trees, because again it's a process in which you can mass produce a large variety, so rapid multiplication can happen in the clonal parks. Okay, and lastly they have controlled conditions, because the environment is controlled. You want to rapidly multiply, you want desirable characteristics, so the whole idea or this part is done through a controlled environment. This is the idea of clonal parks. Okay, now how does this help you in the forest conservation basically? Okay, so one, I think here also the points are good enough. So one is, if you want to conserve the superior genes, this was done in the case of teak, Tectona grandis, eucalyptus, or let's say bamboo species. This was done. If you want to do reforestation, if you want to do it very quickly, clonal parks are very good. Okay, so again restoring of the lost forest can be done through clonal parks. Again, this can help you in the disease resistance also. It can also help you in research activities. Okay, so if there is a plant or a tree basically which is resistant, if you grow a lot of it, again you will be able to have very good disease resistance in that case also, right? Research wise also this can also be benefiting. So it has a lot of importance in the forest conservation, goes without saying. Okay, in India now you should remember the name Institute of Forest Genetics and Tree Breeding. The institute in Coimbatore has developed clonal parks. Okay, so there also they're going for Tectona grandis. Okay, and the whole idea is they're going for superior teak trees which can grow faster, better wood quality, and more reforestation can be done. Okay, so remember the tree species: Tectona grandis, eucalyptus, bamboo. Indian Institute of Forest Genetics and Tree Breeding, which is in Coimbatore, What is the importance in forest conservation? What are the features of a clonal park? Okay. And lastly, what is a clonal park? Okay. So that will help you understand this whole part here. Okay. A similar concept is of a herbarium. Like this is how this looks like. So also here, let's say imagine in a library you have a lot of books. Okay. Similarly, in a herbarium, dried plants, let's say this is a paper or something, a sort of a sheet, herbarium sheet. Dried plants are neatly arranged and they are properly labeled. This is how a herbarium looks like. Okay. We saw the example of British herbarium. Okay. So what is the role? Of course, when you are doing this in taxonomy, the whole idea is classification, identification. So one is identification. Okay. So you have multiple specimens you want to properly identify. It can help you in research. So more you read about evolution, diversity. So yes, that can also be there. Conservation, okay, and education. So a lot of budding botanists, people who are preparing for forestry and other people, you'll also understand about so much things about from a herbarium. Okay. So conservation, and again here documentation, etc. can be properly done. You are properly labeling these things. Anything can be rare, critically endangered. So conservation can also be properly carried out. Okay. So this is the role of herbarium in a forestry. Okay. Here Botanical Survey of India, okay, has a lot of herbariums. So the biggest one is in Kolkata, which has 2.5 million specimens. Okay. It includes very much important species like neem, Azadi Rakhta indica, okay, banyan. Okay, so these are some important things that you should know. Then again here, what is the principles of herbarium? How is it established? This is also important. The principle here will be simple. You will want to document things by collecting, preserving and labeling. Carry out research, okay, which aids in taxonomy, evolutionary studies. This will be used as a reference. Okay, so again a lot of reference species will also be there. In fact, when I tell you reference, I will also tell you this holotype, neotype, lectotype and syntype. Okay, just as reference species. Okay, and again this also plays a very important part in conservation. Again you would see a lot of these points are also repeating, okay, in both of them, because these are similar concepts. Okay, so you don't have to, let's say, think about too many things, right? Right. So it is a very good important source for this, you know, taxonomic information, conservation, reference, research, documentation. How can it be established? Okay, so basically, and again remember there is also one second. Let's say you have a herbarium sheet. Okay, so for this you can just write down a herbarium sheet will be 11.5 into 16.5 inches. Just you can write down the herbarium sheet. Okay. So collection, preservation. Okay, so they are pressed and dried. Again, why dried? Remember we've done this in seed drying to remove moisture, otherwise this will get damaged. Mounting, so they are properly mounted on sheets of paper through glue or tape. Labeling is done. Okay, so scientific name, collection date, location, collector's name, all these things are properly written. Storage in cabinets, shelves, controlled environment to prevent the damage from pests or humidity. That is why they are dried. And last is database management, right? So we want to properly conserve them. You want to have a proper database in which they are indexed and, you know, properly outlined. So they have to be properly stored. Okay, this is through herbarium. Okay. And then the third part and perhaps the last part is Arboretum. Okay, so we have seen clonal park, we have seen herbarium, and then you have Arboretum. Okay, what is that? So here different tree species are grown for study, display and conservation. Okay, just like in a museum you have a lot of things which are at display, here also different trees, shrubs, etc. will be at display for study, conservation, etc. So again, what is the role in forestry? Because these are the kind of questions, roles, principles are asked from you. So one, of course, is conservation, right? Again, I'm not going to elaborate too much here because we've already understood. So they will conserve rare and endangered trees, okay, so that they do not get extinct, number one. Research activities, so again when we see them we understand relationships and studies. Then education is very important to the budding botanists, the people in forestry, etc., and to the general public at large. And lastly is recreation, because it is open, so people can also understand more about these, let's say, trees, varieties, etc., and they can be then be more aware about them. Okay, okay, that is there. Then The Tropical Forest Research Institute, it has a arboretum again, which shows a lot of Indian species. Okay, educational spot for tourists, researchers, and many of the species which are present here. In all of this, please remember. So here I've spoken about Tropical Forest Research Institute. Here Botanical Survey of India, and here I've spoken about Institute of Forest Genetics. Write these examples down or places so that you can differentiate your answer. Okay. Again, what are the principles of Arborata? And again, so here all the four things: conservation, education, research, and recreation. Okay. So CRRR. Okay, CRR you already study in economics. Add one more R. Okay. So that is there. So this is the principles in which this is created, and how is this established? You need to select a site. Okay. Again, a site, soil quality, all these things, water has to be there properly. You have to design this so that different tree species pathways can be there. Planting has to be done. Okay. Maintenance, care has to be done regularly. Pruning, pest control, watering has to be done, right? Labeling, very important, so that scientific names, additional information is there. And lastly is also public engagement. Okay. This is there. Again, see, while this is done, but still there are some challenges also. So challenges I'll also, let's say, mention here. Okay. And these challenges can be common for both, be it herbarium or also, let's say, arborata. Okay. So challenges can also be, let's say, you have lack of infra in general. Some skilled manpower is needed. Maybe you don't have skilled manpower enough. Okay. Again, education and awareness is needed to carry this out properly. Maybe that is not proper. So again, this will not be properly carried out. Okay. So this again is a problem. Then in sometimes you don't have enough scientific labels, so recognition etc. can become a problem. Okay, because of lack of scientific labels or lack of scientific awareness etc. Okay, so this is there. And again, think about arborata, or let's say in these places, maybe they require a lot of land, they require a big infrastructure, and again for that maybe the funds are not available. Okay, funds also for carrying out research, maybe that is not available, the space is not available. So just think in a very practical manner. So sometimes even they can ask you things which you don't have normally read. Again, use your general knowledge to generate points. That is why I'm covering a lot of dimensions when I'm covering the topics with you. Okay, and let us see some of the previous questions which have already come. Here, see, define herbarium, writes its significance. I've already spoken about it. Reference, research, conservation, education, historical record, etc. All of these is a significance. Definition you have already seen. What are the steps in establishing a herbarium? Planning, collection, drying, mounting, labeling, storage, cataloging. Okay, many of the steps are there. And again, just you don't have to even cram them. The moment you understand this logically, you will be able to do that. Okay, and this is the part I was telling you, which is the method of plant classification followed in herbarium. So actually it is the natural method of plant classification. So there also you can mention the Bentham and Hooker model, and even if you can draw that, nothing like it. Okay, then one question was about holotype, neotype, lectotype, and syntypes. Actually, when you are you want to learn about the reference species, then these terms are used: holotype, neotype, lectotype, and syntype. Okay, so what is a holotype? So again, let's say if you've just discovered a very new plant species. Imagine Western Ghats and other areas. Every time a new plant species etc. is carried out. Okay, so the very first specimen that you collect and describe becomes the holotype for it. Okay, so this is actually the official example of that species. Okay, that is the holotype. So number one, this is the original specimen kind of a thing, or an official document related, or official example is the holotype. Okay, now if this gets lost or damaged, okay, so we can select another specimen which can be released. The name is neo. Neo means new. Okay, so this can be done through a new specimen that is known as neotype. Then you have lectotype. Okay, so lectotype, sometimes what happens that sometimes the original scientists, they have described a new species, they've used a lot of specimens, but they did not specify the holotype. Okay, so then one of the species or the specimens can be used as a lectotype, which then becomes the main reference. So you have holotype, the original specimen. If this gets damaged or lost, you create a neotype, and sometimes you use some other specimens without specifying the holotype, then you can create a nectotype. And last is the syntype. Okay, So here multiple specimen, okay, so can be used. So all this collectively can become a syn type, okay. So for the same species, if you use multiple specimen at different places to tell or let's say educate or organize about it, that is one thing that you can understand, okay. Now second part, describe the role of arborata and clonal plaques in conservation of biodiversity. We've already seen that. Arborata, okay, so it is just like a botanical garden. So it is going to conserve endangered tree species. Again, I gave you these important examples. Tropical, let's say, Forest Institute in Jabalpur. Okay. Similarly, I gave you the example of Institute of Forest Genetics in Coimbatore, in clonal parks. How they play the important role. I've added 1 or 2 points, but you can add many points. We've discussed this. Okay. So I don't think this is a difficult question to answer. Okay. Arborata, clonal parks, etc. Right. Again, a question, for example, define ecological amplitude. We have seen this in the previous lecture, that it is the range of environmental conditions within which the species can survive and reproduce. We are talking about an entire range. Okay. What are the characteristics, or let's say classification of plant communities and characteristics? Here you can definitely outline the different classifications we have studied. Okay. And for the characters of a plant community, you can think about species composition, okay. You can think about the dominance, you can think about diversity, okay. You can think about the structural organization in terms of canopy. And by the way, all these things I've also already explained to you in the previous lecture. The different terms, remember the forest community in that characteristics and characters, okay. So these are the characters, right? Successional stage, functionality, okay, appearance, or the general structure. So all these things are not that difficult, I would assume now, for people here, okay, in this class at least. And again, let's say classification can be based on dominance. So you can have a monodominant or a polydominant community, xerophytic, mesophyte, hydrophytes. You've seen this in one second, in ecological succession also, okay. Successional stage, pioneer, climax, based on habitat, terrestrial, aquatic, based on altitude, okay. So these are few things which you understand here, okay. Again, I'm telling you, here you have to slightly focus more on the taxonomy part. You go through this once, because arborata, clonal plaques, herbarium, these are easy. But maybe you have to get a hang of how to write the initial dendrology and taxonomy part, because classification can be new to some of you. Other than that, this is again a high yielding portion, okay. So this basically brings me to the end of this lecture. And again, in the next lecture, we will see the ethnobotany, which again is a very important part here, okay. So we started with dendrology, we saw some case studies, we saw taxonomy, we saw classification, we saw different kingdom types, okay. We saw Raunkiaer's life system, Linnaeus, Bentham-Hooker, Eichler system, okay, different kingdom classifications. Then Eichler system, Hutchinson systems, then the different taxonomical aids for clonal plaques, let's say herbarium, arborata, okay. And then we ended the discussion with some previous questions which have been asked in your exam, okay. So now I'll meet you in lecture number 3, which is related to ethnobotany. Thank you.